Hey, biology class. Welcome to our first set of notes here for chapter 13. We're going to look at genetics, which if you took Gen Bio, I think you're going to fly right through this. So that's my hope. Inherited traits, genetics, it's a pretty fun topic, and it's really not too terribly difficult. So your chapter starts out with kind of an application story, talks about cystic fibrosis. Cystic fibrosis is our most common fatal genetic disorder in the U.S. It is pretty common. The CFTR gene encodes a CFTR protein. All right. This CFTR protein is a transport protein that moves chloride ions out of the epithelial walls. And when you move chloride ions out, water follows that by osmosis. And so what you end up getting is epithelial cells have this thin layer of water on them, and they are basically lubricated all the time. And that's a good thing. You want that in your epithelial cells. Now, if you have cystic fibrosis, you've had a deletion of a few base pairs, and what you end up getting is this CFTR can no longer make it through the membrane. And if it can't make it through the membrane, it can't transport chloride ions. And if it can't transport chloride ions, your epithelial cells have a dry surface. Now, this is especially a problem in the trachea and the lungs. Mucus is something that gets in your mouth and in your trachea all the time, and it's not a big deal. Cells get rid of it, it moves through the system, that kind of stuff. Cells can bring it in. It's a normal part of the digestive tract. It's a normal part of an esophagus. It's all over the place, right? Mucus will stick to the epithelial cells in the lungs because they're so dry. What would normally slide off now gets stuck. And when it's stuck, it allows bacteria a good habitat to hold on to. The other thing that CFTR does is it plays a big role in identifying bacteria and having cells undergo endocytosis to bring them in and kill them. And so once a person has cystic fibrosis, not only do they have mucus in their lungs, which is a great habitat for bacteria, but they also aren't able to identify those bacteria quickly. And so what we end up getting is just this perfect storm of having a habitat for bacteria, not being able to kill them quickly, and you basically have what would be like a never-ending pneumonia. And so we give them an antibiotic, and the bacteria come back, and we give them a stronger antibiotic, and after some time the bacteria come back, and we give them a stronger antibiotic, and then the bacteria come back, and their mucus this whole time is building up more and more and more mucus in their lungs, and it's going to be fatal 100% of the time. It's going to be fatal. We have stretched this lifespan from what started off to be maybe in the teens all the way up to 30 years old, which for a cystic fibrosis person, that's really old for cystic fibrosis. So we've come a long ways, but it is always fatal. So little changes, little tiny changes. I mean, that's 3 base pairs. That's not a huge change. That could have, by dumb luck, that might have been, those might have been redundant and coded for the same protein and the same amino acids, but they didn't. And so now we end up getting this deal that's going to kill you before you're 30, even with constant medical attention. And so here's the protein, the CFTR protein, and that little teeny tiny green strand right there in this whole complex shape, that little tiny deal is all that was changed, and this whole mess follows. So protein production and protein regulation are incredibly important, and you guys already knew that from this class, but now we're going to talk about the genetics and how you get this information and how this leads to this phenotype that we're talking about. So when we look at genetics, we talk about a guy named Gregor Mendel. You don't get very far in genetics without hearing the name Gregor Mendel because he's called the father of genetics. So way back in the 19th century, we knew that kids somehow inherited information from their folks because the kids look like a blend of mom and dad. We just didn't know how it worked. They thought maybe hereditary material was some sort of fluid that was all blended together when kiddos were fertilized, but we weren't really quite sure how these two traits would blend because we didn't always get a perfect blend. You cross a black horse and a white horse, you don't get a gray horse. You might get just a black horse, or you might get a black horse with white spots, or you might get, a black and white paint horse. There's all these weird kind of blends, and so it wasn't really working out as well as our simple explanation that we would like. You guys can probably see this in your family. You might have a mom and dad with brown hair, and you might have a blonde-haired sister and a brown-haired brother and a red-haired, brother or something like that. It's just not always this perfect match. And so Gregor Mendel was really, he wasn't the first guy to study this necessarily, but he was the first guy to study it and get good results and publish it. And this groundbreaking, amazing discovery sat unrecognized and unappreciated for a long time, till like after his death, before somebody finally found his manuscripts and figured out that he had pretty much cracked the code to, I don't know, like all the information that makes us us, right? So he's a monk, and he was good at plants, and he was good at math. And so he took garden peas, and he noticed that they bred true for traits. Like he would have peas that just had white flowers, and all their kids would have white flowers. And he'd have peas that just had purple flowers, and all their kids would just have purple flowers. And so a true breeding trait appears the same in all the offspring as it does in the parents. And that can be kind of hard to find. So just finding that was pretty good. So he discovered that these traits of offspring in these cross-fertilized pea plants appeared in predictable patterns. So what that means is he took all his purple flowers and all his white flowers, and he bred them across. A purple flower bred with a white flower, and another purple flower bred with a white flower. Plant all the seeds, grow them, and see what they look like. How many are purple and how many are white? And because he was such an awesome math nerd, he started to get some pretty predictable ratios that he thought, hey, there's some information here that is being carried on in a predictable amount. And so that was kind of cool. So he concluded that this hereditary information was passed from one generation to the next in discrete units that we could measure mathematically. We didn't know what DNA was. We wouldn't know what DNA was for almost 100 years. We didn't know what genes were. We don't know what alleles are. But with some math and some time, this guy figured it out. He's so far ahead of his time that nobody knows what he's talking about, because that's what happens when you're really, really smart. The cool thing about how super smart he was is his experiment's not that complicated. That's a pea flower. That little guy is called a carpel. It is the female reproductive organ of a flower. That little guy is called the anther. That's the male reproductive organ of the flower. Cool thing about a flower is, for lack of a better term, they're really easy to castrate. All you do is you grab these little yellow fuzzy guys right here, and you just cut them off or tear them off with your hand. And you just took a hermaphroditic, perfect flower with male and female parts, and you just turned it into a girl only. It's that easy, real quick and easy. And so here's what you do. He grows this yellow plant, and as soon as it starts to grow its anthers, he cuts them off. This yellow plant can no longer self-fertilize. He can't make pollen, but this purple plant can. And so you take this little yellow, fuzzy pollen that everyone's sneezing out in the spring and summer, and you put a little bit of it on a Q-tip or something like that, and you put it on the end of the carpel, called the stigma, the end of that little stigma on the carpel, and you have cross-fertilized a purple flower with a white flower. And you know you've cross-fertilized it, because if this guy produces seeds, which he should, his peas that he produces are full of embryos that could not have come from these anthers. They had to come from these anthers. And so we get a guaranteed mix between purple and white flowers. And so you grow them, and you get a whole bunch of purple flowers and probably a couple white flowers. And you just count. This plant produced purple flowers. This plant produced purple flowers. How many plants produced purples? How many plants produced whites? And he did this with more traits than just flower petal color. How long the pea pods were, how tall the plants grew, what the actual peas were like. Were they wrinkled or smoothed or yellow or green, all this kind of stuff. He did a lot of experimenting with peas. And it was a simple design, and it worked. So in modern terms, he can't call it this. He had to call it something like, what do you call it? Like an inheritance factor or something like that, because he was just using numbers. But if we were to say this in the terms we know now, each gene has a specific locus or location on its chromosome. And so there's a gene that codes for the color of the flower petal. And there's a gene that codes for a pea pod being green instead of yellow. These are all different genes. And the set of alleles that the individual carries is that genotype. So an allele is a form of a gene, an alternative form of a gene. And the sets that you carry are going to determine your genotype. Your genotype is your DNA. The DNA, the set of genes that make you, well, that's a tough thing to say, kinda make you you. We'll talk about that more in class. If you have two identical alleles, you are called homozygous. If they are different alleles, you are heterozygous. A lot of the traits expressed in you and I, and in animals as well, are heterozygous. Are just two different forms of the alleles can account for pretty much all the phenotypes you're going to see. Oops, I threw that word in there without telling you what it was. Sorry. A phenotype is what comes from your genotype. Your genotype is your genetics. Your phenotype is the way you look. Hair color, eye color, height, that kind of stuff is the phenotype. And so here's an example of some loci or locus, location. So here's chromosome 7. We got a set from dad and a set from mom. Here's chromosome 15. Here's a set from mom and a set from dad. And here they have the same genes in the same location. They've got genes that produce the protein elastin, and they've got homeotic genes, and they've got genes that make them colorblind or not colorblind. Here's a gene that can cause Tay-Sachs disease. Here's a gene that's going to code for how much pigment's in your skin, right? And they line up in what we call homologous chromosomes. So you have one copy of that gene from your dad and his allele that he passed down, and one copy of that gene from your mom and her allele that she passed down. And depending on which of those is expressed and how much of it is expressed, that can make you colorblind or not colorblind, and all sorts of different stuff. So in modern terms, a hybrid is the heterozygous offspring. So a hybrid is a heterozygote, which means it has two different alleles. It's normally a cross between two true breeding individuals for the mom and dad. Its genotype determines its phenotype, more or less. You can certainly do some things to change your phenotype, dye your hair, wear contact lenses, exercise, not exercise, that kind of stuff. But a lot of it, all of it, is going to start with your genotype. And your phenotype is your observable traits. The stuff I can see. I look at you in class. I don't know what your alleles are, for hair color, but I know your hair is mostly brown. That's observable. What alleles exactly make that up? I don't know. It could be a combination. I don't know that. But I know your phenotype. I can see that. So if you mutate a gene, you have a new allele. That might affect your phenotype. It might not affect your phenotype. But if it's a mutation, it's unique now. It's different. And so that is a new allele. And those certainly can change the phenotype, like you just saw with cystic fibrosis. So, an allele. You hear these terms dominant and recessive a lot, and I don't know if they're really always used correctly, but here you have it. If you have a dominant allele, it masks the recessive allele. And so you can have a recessive allele and not know it because it's not expressed, but it's there in your genetic material. You can pass it to your children. You just see the dominant allele because it's expressed in your phenotype, okay? This might be a little too confusing, but I am A positive blood type. My mom had A blood. My dad had O blood. I'm A positive blood. Now, my dominant allele for that is A. My recessive allele for that is O. And actually, they're co-dominant, but if you look at my blood donor card, it says A positive. You don't know if I carry an O allele or not because you only see the A. Okay? And I do carry the O, but that's the difference between phenotype and genotype. Not everyone knows their genotype, but in my case, I'm lucky enough to know that. So that's one example. The other example could be brown-haired mom or dad that carries a recessive allele for blonde hair color. And you look at them and they have brown hair, but then they have this kiddo and their kiddo's blonde. Well, they carried that gene, you just didn't see it because it was masked by the dominant brown gene, right? So that's another example. We use capital letters to signify dominant alleles, and lowercase letters signify recessive alleles. And so if we were homozygous dominant, it'd be big P, big P. If we're homozygous recessive, little p, little p. And if we're a heterozygote, we are big P, little p. All right, so that's the way you can do this. And we'll do some Punnett squares and check those out. And so here's the P-plant example, right? Homozygous dominant, big P, gives you a purple flower. Little p, gives you a white flower. Big P, little p, heterozygous, gives you a purple flower. Now, if you just see the flower, you don't know that he's carrying a recessive allele because he just sees the purple flower. But he is carrying a recessive allele. These two guys look the same on their phenotype, but their genotypes are different. And there are some experiments you can do to figure these out, and that's what Mendel did. But just know that the genotype determines the phenotype. But you can't always take a look at the phenotype and know the genotype from it. It takes a little more work than that. So I guess in this example, it'd be important to note that purple is dominant to white. If both of those alleles exist, the purple is expressed and the white is not expressed. That's called simple dominance. All right, so that's 2 sections of chapter 13. We will stop before we talk about segregation, and we will save that for the next video. So thanks for your time.